In this video, I'm going to go through with you the required practical for chromatography, required practical 7, A-level biology, and I'm going to be doing it with TLC paper. So I'm going to go through with you the equipment, the method, key mistakes students might make for you to look for, but also emphasising what are the common exam questions that come up linked to this, so you can make your students aware of that as well. And if you want all of that bundled together in a free guide, that is linked in the description for you. Don't forget to hit the subscribe button so you don't miss out on any of these required practical videos. And a big thank you to Woodford County High School for letting me film all of these videos here. So the aim of this experiment is for the students to be able to separate out the pigments and to know that the reason that they separate out is because they have different solubilities. And depending on the solvent that you have, you'll get a different number of pigments because the pigments may be more or less soluble in certain solvents. So they're going to then create their chromatogram, calculate an RF value, and then they can use that to compare different leaves, or they can use it to identify the pigment that they have. I'm going to be comparing 2 different leaves. Whatever you have available, you can choose to compare. You might want to do a shade tolerant versus a plant that isn't tolerant to shade to see if there's any difference in the type or proportion of those pigments. So the equipment then that we have for this experiment is I've got my TLC paper, I've got a ruler with millimeter measurements and a pencil. I've got capillary tubes just here. I have my organic waste bottle to dispose of the solvent safely at the end. Forceps, hairdryer to help dry the pigment if we need that. And then I've got in our tubes here, this was the spinach which has been mixed with the solvent already to get the pigments out of the leaf. I've got my coleus here, which is another leaf which again has been mixed with the solvent. And for that one, the solvent that was used to extract the pigments was ethoxyethene, and that was mixed with propanone in a 1:1 ratio. Then I've got 2 McCartney bottles. Both of these are the running solvents inside that I'm going to put the TLC paper inside of. And the solvent that I've got there is cyclohexane to ethoxyethane and to propanone, and that's in a ratio of 5:3:2. The main thing here of the risk assessment is the solvents. So these solvents are volatile, they're flammable, and they give off really, really strong fumes, and it will cause severe headaches and potentially dizziness. So you either need to be working inside of a fume cupboard, or we've got all of the windows open here to reduce the chances of headaches and any issues linked to the fumes. So now we're ready to get on with our method, and I'm actually going to run 2 TLC plates, 1 for each pigment. TLC paper is expensive, so you can make this cheaper by cutting these in half lengthways, because that is still wide enough for you to be able to get a good chromatogram. And what I then need to do is draw my origin line in pencil, and then I'm going to start adding the pigment. But I need to work out at what distance up to draw my solvent front. So what I'll need to do is measure how far from the bottom up the solvent is, take that bit of glass distance away from that to know that is how deep in terms of in the bottle the solvent is, and that is where I'll draw my origin line. So it looks like 6 millimeters, and that is 18 millimeters. So 18 minus 6 would be 12. So they just need to dip it in, keep your fingers over the tops, creates a bit of suction, dip it on the paper, but you need to wait until it is dry before you then add the next drop, which is why I've got the hairdryer here, so you can see when it is then dry. And we'll keep going until we've done 10 drops, or at least 5 millimeters in diameter. They probably won't need to dip it again, this one. And wherever possible, closing the lids, because that's going to be letting out the solvent. So I've now got my 2 TLC papers ready to go into my 2 separate bottles of solvent. I've got my origin line drawn, I've got my pigment above where the solvent is going to reach, and then I've got my pencil and ruler at the ready to take it out afterwards. So we need to make sure that we are lowering it in vertically. And I can see that the solvent was below the pigment line, so that's good. Right, so the one on the left is almost halfway done already. The one on the right, my pigment has gone slightly below the solvent line, so it might be an issue, although both of them now it looks like a green one starting to emerge from the top. Actually, this one, yeah, you can start to see a couple of coming off the top. So I'm going to take mine out now before it reaches the top. I'm going to do it one at a time. So I can draw that solvent front on straight away. Now if we just zoom in and have a look at this one before I get the next one out. So I'd still work out the RF values for those and then look at which pigments would move or have that RF value in that particular solvent. So that one was my spinach. So I'll do the same and take out the next one. Drawing on that solvent front straight away before it evaporates. And I'd get the students to take a photo at this point before any of the pigments fade. And then what I can see is we've got a faint yellow one here, and then I've got a dark grey, green, a yellow. There's 3 distinct other ones as well that I would measure the RF values for. So once the students have then completed their experiment and they've got their chromatogram, take a photo immediately, because the solvent front will disappear as the solvent evaporates, but also the pigment colour can start to fade over time. So they can always go back to the picture, plus it's nice for them to stick that in their lab book. What their aim is though, is to be able to identify the different pigments and try and work out which pigment they have based on the calculated RF value, and then they can research for the solvent that you used, for the RF value, which pigment is that, especially if they've got the colour as well to help back that up too. So some of the key things to point out to them: if they've got pigment spots where it is spread out, they should be measuring from the middle, the centre each time. So that is an average, and also they're standardising how they're measuring it. So they need to record from the pigment point on the origin line to the middle of each pigment, what is the distance that the pigment moved, and it'd be best to do that in millimetres to be more precise. They should also be measuring the solvent front. So that is where the solvent was initially, where the solvent ended, and that is what they're going to be dividing by each time. So it's the key things to make your students aware of then. They should always be drawing in pencil, because if they use ink, the ink is going to be soluble and that will move up as well with the pigments. And that is a common exam question for 2 marks. They should also make sure that when they place their TLC paper in the solvent, that the solvent lies below where the pigment is. Otherwise, it could splash onto it, dissolve the pigment, and then you lose your pigment spots. Again, that's a common exam question. They need to make sure that their TLC paper is straight and not at an angle, because otherwise as the pigments move up, they might go off the edge of the paper. Another common exam question is that one. They have to keep a close eye, because when the solvent does reach just before you hit the top of the paper, that's when they take it out, so that you don't get all of the pigments reaching that same end point and you don't have them separated. As soon as the students have seen that the solvent has reached just before the top, take it out and they need to draw a pencil line on immediately. That That's because the solvent's going to evaporate really quickly, and as soon as it's evaporated, they won't know the point that it went up to, and then they can't measure the solvent front. They should also make sure they're not putting their fingers on the TLC paper at all, so that can interfere with the ability for the pigments to move up and affect the results. So that is it for required practical 7, and don't forget you can get the entire guide for this practical for free, linked in the description, and hit the subscribe button so you don't miss out on any of my future videos.